Hello, everyone. Welcome out. How are y'all doing? Con going well so far? All right, welcome out to When Genes Go Rogue. So I will be your moderator, Jason Wallace. I'm joined by my four lovely panelists who will be introducing themselves shortly. To start off, we've got a few administrative things. First, please silence your phones. We're sure you have a lovely ringtone, but now is not the time to share that. You can do that in between panels. Let's see. You've probably heard that we are supporting the charity NAMI, National Association for Mental Awareness for Mental Illness. The QR code's up here. I can't see it, which is why I have trouble with the acronym. But please go ahead and donate to that if you have not already. If you're looking for more science track stuff after this, next one in this room will be at 5:30, which is the science of propaganda. And then at 8:30 tonight in the ballroom, we are going to have silly science with remarkable results. So with that out of the way, yes, and we do have people trickling in. So again, please make friends with your neighbors. If you see seats next to you that are empty, that are in the middle, and they're gonna make people have to climb all over you, just scoot over a little bit. 1, 2 seats, it's fine. Open up spaces for people coming in. So with that, let's get down to talking about rogue genes. So like I said, I am Jason Wallace. I'll be your moderator. I am an associate professor at the University of Georgia, and I study corn and microbes and how we can get them to work together. And I also, with that guy down there in black, co-host a podcast called Gaming with Science about board games and science. And there's ribbons in the back if anyone wants to pick some up. And now, my lovely panelists, will you please introduce yourselves?
Hi, everyone. My name is April Arms. I see a lot of familiar faces from the last panel I was on, so welcome back. I am a microbiologist. I got my PhD from the University of Tennessee. Go Vols. And I studied the environmental side of microbiology. With that, I studied how microbes, particularly bacteria, interact with one another and how they can talk to one another. Yes, bacteria talk to one another. And so I am what someone earlier had said, a recovering scientist. I know I'm no longer in the lab, and now I like to say that I'm a professional networker. So I help faculty connect across the institution and outside of the institution.
All right, and I'm Topher. Hello, I'm Topher Hunter. I started out as a PhD student at Tech, doing bioengineering, regenerative medicine. From there, moved on to faculty at the University of Calgary, and then moved back to industry. And I've spent the last decade and a half working in infectious disease products as both development and as a professional communicator. I go literally into the operating room and talk to surgeons and help them use our products and stuff like that.
Hi. I'm Brian Kvitko. I'm an associate professor in the Department of Plant Pathology at the University of Georgia. So we study diseases of plants. I do a lot of work with Vidalia onion and its diseases, the bacteria that cause disease on it. Let's see, what else? I am still a suffering scientist. I am not a recovered scientist. Our moderator, Jason, mentioned we do a podcast on science and board games. So.
Hey, I'm Jen Hardee. So I'm originally a local girl. I'm from Tuscaloosa, Alabama. Roll Tide. So I got my PhD in molecular biology and human genetics at Yale, and now I work out in the San Francisco Bay Area at a pharma company. And one of the things that we work on is bioengineered T cells for cancer treatments. I've also done some work with CRISPR, and I've got a couple of CRISPR patents, so I'm sort of holding down the human side.
All right, thank you very much. So let's start off. So those of you who were in our earlier panel on microbes know we spent about 10 minutes talking about this and could not arrive at a definition of what a microbe actually was. Now, genetics has been being studied for 100 years, so I'm sure...
Why are you doing this, Jason?
Surely someone has come up with a definition we can actually use. So panelists, can you tell us, what is a gene?
Sure. Let's see. The problem with all of this is it's hard to find a definition that we feel like fully encompasses everything. But the easiest definition that's going to work for most things is that a gene is a stretch of DNA that is translated and made into a protein that actually does some kind of a job. I say that's not everything, because there are also genes that when it gets turned into RNA stays RNA, and that piece of RNA is what actually does the work. So I think for the most part, that's not terrible, right? Is that reasonable? Okay,
Yeah,
I'm getting some not so much. So people are going to elaborate on that definition.
Yeah, I would say it's good to think of genes are a blueprint. You know, genes typically don't do anything in and of themselves. They are the instructions for making the widget that then goes out and does the thing.
Mm-hmm.
Yeah, I have written down here in my notes that I would define a gene as a section of DNA that encodes something. And I generally like to think of genes in terms of bacteria. So what's interesting is that, you know, bacteria have these genetic coding regions that are pretty much like all of bacterial DNA is a genetic coding region. And which allows them to be very efficient at the things that they do. However, I spent a postdoc in a plant synthetic biology lab, and I was constantly having to be reminded that plants have introns. They have these areas of non-coding regions of their DNA, and that are not genes, do not encode anything, and that would always trip me up.
Yeah, you guys have probably heard about junk DNA and how, you know, the human genome is 98% junk DNA. And I think junk is the wrong word. You know, genes get all the glory, and we're going to spend some time, most of our time on genes today. But, you know, it's like your attic. It's not full of garbage. It's full of interesting stuff you salted away because you might need it sometime.
Yeah, bacteria still have a better solution to this, though. they're just information dense little packets, right? It's like everything's just there. There's so little room for waste. Like if you've got a stretch of, like, your average gene is maybe 1,000 A's, G's, C's, and T's, right? And for so in 10,000 bases of bacterial DNA, you can have 10, 20 genes potentially, and all their regulatory elements and everything. If you look at your DNA, they're like weird little islands. There's like one gene, and then there's nothing. And then even in that gene, half that gene gets thrown away. It gets turned into RNA, clipped out, and I don't know. Eukaryotes drive me absolutely crazy.
Oh yeah, look at Mr. I don't need a nucleus.
All right, so we seem to have actually settled on a decent definition of a gene. The whole panel is about genes going rogue. So what do genes do normally? And most importantly, what does it mean when we say they're going rogue?
So as I was saying earlier, genes are really the blueprint for making something. And I think a lot of people get this wrong. So it's like, all right, where are your genes? They're on your chromosomes. Where are your chromosomes? I think a lot of people think, okay, they're in eggs and sperm and all that stuff. Every single cell of your body has the same DNA, every single one. So what makes each cell different? Why is one a lung cell and one's a pancreas cell and one's a hair follicle? That comes down to the expression of your genes in each cell. So I'm a musician, and the best metaphor I've seen for this is think of genes as the instrument that you're handed, your keyboard, your guitar. So you're limited by what the instrument can do, but what then happens in terms of the genes expressing and doing things is the music that you choose to play. It's how long, how loud, which note, when do those things happen? And so that's how genes that are all the same in every cell of your body then turn into, I'm cold today. I should do something about that.
I like that you brought up chromosomes. So in eukaryotic organisms, most of the chromosomes are linear. In bacteria, most of the chromosomes are circular. But not only do bacteria have these circular chromosomes, which largely encode most of the proteins and have most of its genetic coding region, but we also have plasmids, which are extra chromosomal Basically, you can think of them as tiny mini chromosomes. They're also circular pieces of DNA.
Yeah, those things in Bioshock were real.
All right, so you keep throwing around the word like prokaryote and eukaryote. Can we do a quick definition of that?
Sure. Okay, so first of all, we've got 2 bacteria people here, April and I, and then we've got 2 we're not bacteria. This is not the meaning of the conversation. So you and almost every single thing that you can see are eukaryotes. That means you've got large cells that have a nucleus that holds all of the DNA. And then most of the living things actually that you can't see, and actually most of the living things both by diversity and number, are prokaryotes. Those are bacteria, those are archaea. They don't have a nucleus. Their DNA is sort of semi-sequestered inside of the cell. I think that's pretty good. Basically, that's the biggest thing is, do you have a nucleus or not?
Yep.
All right, so how do genes go rogue in both those systems?
Okay, so I could talk about bacteria things. So when I'm thinking about when genes go rogue, right, what's supposed to happen? You have genes, you pass them to your offspring, right? That's how this is supposed to work. They go, that we call that vertical. Like if I have kids, they have my genes. When bacteria divide, they're going to make clones, right?
Sure
one cell splits into 2 daughter cells. They should have exactly the same genes in it, right? That's vertical inheritance, and that's normal. We do not consider that going rogue. Rogue is like these plasmids thing. Rogue is when genes move laterally, not from child to offspring, but from here, April, have this gene.
Yeah, so we call these like mobile genetic elements, and there are several examples of mobile genetic elements. So plasmids being one. There's also transposons, which are just small regions of DNA that literally like hop out of that section of DNA and then go somewhere else. And it could be within the same chromosome, or it could be somewhere else. Then we also have viruses that are specific to bacteria, that bacteriophages that will sometimes accidentally package bacterial DNA. And that allows that DNA, that bacterial DNA, to then go and be transplanted into a different organism. And we've also got horizontal gene transfer, conjugation, which is kind of like it sounds. This is not a 10 p.m. panel, so I'll put it in more scientific terms. But one of the bacteria will form a pilus, which basically injects DNA into another receiving bacteria.
Yeah, that's one of the ways that antibiotic resistance can get spread from bacteria to bacteria. They can basically hand it off to their friends and be like, hey, look what I found.
So bacteria produce clonally. That's what they do. They are supposed to divide, they make perfect clones, so they don't have to mate to reproduce. And for things that don't mate to reproduce, man, they still share genes a lot. And they share genes using these horizontal transfer mechanisms. They have plasmids that can move around. Now, the idea that the bacteria, it's making a little pili, that it makes a little channel, a bridge to send itself to the other cell. It's not really the bacteria that's doing that. The genes that let you do that are on the plasmid itself. The plasmid is coding for all the genes to maintain itself and to move itself from bacteria to bacteria. Viruses can do that too. Those are mobile genetic elements, but bacteria also eat DNA. As you would, right? It's a valuable resource. It's expensive to make.
It's a nitrogen source.
It's a nitrogen source. It's a phosphorus source. It's a carbon source. So if there's just DNA sitting out there, there are bacteria that have systems to take that up. And mostly they're going to break it down. Sometimes they don't break it down. Sometimes they take that naked DNA out of the environment and they recombine it into their own genomes. And at that point, you can literally get a chunk of anything.
Yeah, I think it's worth coming back to something that April was just mentioning, that when a virus attacks a cell, so the virus has no ability to actually reproduce itself. It can't reproduce sexually or clonally. It injects a sequence of DNA into the host cell. So you get COVID. COVID injects its DNA into your cell. It co-opts, so it takes over and uses the replication machinery that your cell has to make new copies of itself and make new the coat, the plasmid coat, or the
Envelope.
thank you, that envelops the whole thing, and then that makes new viruses. So it's borrowing everything. It's like a stranger coming into your office and taking over your Xerox machine, and it's going to make a bunch of copies for itself, and then it walks away. Well, if you accidentally
it blows up your room.
Sometimes the room blows up. But what if you had left, a document on your Xerox when that person comes in? Well, now they're going to lift a copy of that.
Now it's got my driver's license. I'm not cool with that.
So that it becomes a part of them. So the virus has now sucked up a piece of the host DNA and it's incorporated it. And if it's a viable new piece of DNA, then that virus is mutated and adapted.
This thought just occurred to me that I feel like this means that all viruses are just rogue genes.
Yeah, fundamentally.
Yeah, I mean, kind of. On the human, ukes side of things.
Prokes.
Ukes, baby.
No, don't guys. No.
So we have a couple of different ways things can go sideways. First of all, I think is the thing people are most familiar with, which is genetic mutations. And those can arise a lot of different ways. Sometimes it's just you have lived a full life and you went to the beach a few times and you ate and you drank stuff and base pairs get tweaked. And so that changes your underlying instructions for how things get made. Most of the time, it doesn't change much that matters. Every once in a while, it's going to hit something important. The other thing that can happen is that, how I was saying genes get turned on and turned off, and there's this sort of the, music that you play to make the proteins when you need to. That regulation machinery can get mutated. So basically, if there's often an element right upstream of human genes that tells them when to turn on and when to turn off. You can just break that so that they're always off, always on, and you can see how both of those could be bad things in various contexts.
And that's where you get things like virally induced cancers.
Yeah, that's no good. So virus, inserts itself into your genome. It's usually random. And if it happens to hit one of those regulatory elements, it means this cell should grow all the time, pedal goes to the floor. That's an easy way to turn into a cancerous cell.
All right, so you mentioned a while back these transposons, these jumping genes, which I have heard are actually active in my own DNA and jumping around right now. Is that true? And how much do I need to worry about it?
I'm a bacteria person.
I mean, we have transposons are everywhere. Transposons are fundamental. Bacteria have them too. But again, like I was saying, your average eukaryotic cell, a huge amount of the genome is bits and pieces of busted transposons. I don't know why bacteria have done such a better job of keeping this under control. They've been around for longer, so you'd think it would be more of a problem.
Yeah, we've got a ton. This is what a lot of that junk DNA that I was mentioning earlier is leftover bits and pieces of transposons, bits and pieces of viruses, and it's mostly just sitting there not doing much. Occasionally it does get to hop somewhere else, but again, it's got a 98% chance of hitting something that doesn't matter. So for the most part, transposons aren't really very active in people, and when they do move around, your odds of it causing a problem are pretty minimal. So I wouldn't get too excited about it.
I wonder if one of the reasons that bacteria are more subject to transposons jumping around is because they evolve at a much faster rate than we do as humans, and that might be one of the reasons why we're not seeing as much activity in our own DNA as we do in
one
bacteria.
Thought experiment I've been wanting to do. I've never managed to do this. Maybe Jason or somebody else can help me figure out how to do this. Corn, maize has a lot of transposons in it. And I've always been curious, in your average bowl of cornflakes, by weight, how much is transposon?
I feel like this is asking what percentage of my pillow is dust mites by weight. I don't think I want to know the answer.
Just don't, no.
All right, so we've mentioned a few times bad things happening from genes going rogue, cancer being a big one. Are there times where it's beneficial, or at least from our point of view, beneficial? It's that gene has gone rogue and we consider that a good thing.
Oh yeah, totally. So my personal favorite one, anybody here like cheese? Lactase persistence, baby. So in most of the world, when you grow up and you stop needing to drink your mom's milk and you get onto solid foods, your body turns off the gene that you need to digest lactose. Well, in Europe, there's a whole mutant strain of us where it doesn't turn off and it stays active into adulthood. I love ice cream. I like my rogue lactase persistence. Thank you very much.
So we just talked about this in an episode of the podcast. I don't remember. I think we were talking about Mendel's peas, and we got onto this topic of lactase persistence.
Do you know, was that done by like a transposon jumping in, or do you know what caused that change?
No clue.
Okay.
Do you know?
No.
Okay, I don't either. But you said that it is a huge benefit, right? In terms of its advantage, you said that it's like 4 or 5 times as many offspring from lactase permanence or lactase persistence if you have it versus you don't.
In a population where there's dairy to drink.
Of course.
Provides
Otherwise,
no
it's not going to
benefit
do you any good.
Yeah, there's some other fun ones like blonde hair. There's a lot of traits like that are mutations. You know, most people would have dark hair and dark eyes, but then we have a handful of weird random pockets where...
Hey.
I've got purple hair. I have no room to talk right now. So yeah, blonde hair, red hair, green eyes. There's a whole pocket on the Solomon Islands which have completely separately developed a completely different blonde hair gene. So this kind of stuff pops up all over the place, and it's really cool. I think it's how we keep humans neat.
So I think there's a lot of examples of both like good and bad genes going rogue in bacteria. Of course, spreading resistances. Sure, you guys have heard of antibiotic resistance, right? And that's spreading in bacteria, bad. However, there's also spreading of heavy metal resistance, and spreading of degradation pathways, which can be good for environmental systems and remediation purposes.
Oh, these are like the ones that can live in the Chernobyl reactor?
Yeah.
There's some probably it would take us off topic too much to go into it a lot, but there's some fascinating work in environmental remediation for taking these naturally identified mutant bacteria and utilizing them, like finding what was it? There was recently one species or strain of bacteria that can, I might get it backwards, but it can reduce uranium from a water-soluble to a water-insoluble oxidation state, which means in water-soluble, it goes everywhere, and we get, you get radiation problem. In the insoluble, it just sits there, and it's really easy to deal with in a cleanup. So, and it's a bacteria that does this. It's like it's eating uranium and converting it. It's so cool.
Yeah, bacteria for the win.
I'm a fan.
All right, so getting back to rogue genes. So you've mentioned a few ways that they happen. They can be good, they can be bad. It sounds like, at least for most of the ones that are inside my DNA, I would consider them bad. The genes may disagree, but I think they're bad. But I'm also guessing that the evolution has made some ways to deal with them. So how does evolution keep genes from going rogue?
A big thing that we have in like eukaryotes is DNA checkpoints when the cells are dividing. So there's a bunch of genes that are responsible for basically saying, hey, okay, I'm getting ready to divide. Is my DNA in good shape? Yes, no. And they actually go around and they check, and it's even easier with us. We've got pairs of everything. They can be like, do my chromosomes match? Are we good here? And if it finds problems, it fixes them. And then, all right, now we can move on to replicating and making a second copy. And then there's another checkpoint where it sanity checks. Does what I just made match what it came from? And so that's a big control mechanism that's in all of our cells. And there's a lot of different checkpoint genes. There's a lot of different DNA repair mechanisms. This stuff is going on all the time.
But some of those mechanisms are sometimes like, close enough.
Yeah, and that's when things go wrong. So typically, in order to get, say, like cancer is the one everybody thinks of, right? You usually have to have 2 different things go wrong. You have to lose the brakes, and you have to have the gas pedal 100% on. So you'll have cells that sort of slowly accumulate mutations over time, and if you happen to get one of each of those in the same cell, then you can get cancer. And then that's the kind of thing that also is where hereditary cancer syndromes come from. Like if something runs in your family, like breast cancer, it typically means you have one bad copy of often a DNA checkpoint gene. And so your cells aren't checking as thoroughly as they ought to. Or you have one bad copy of a growth gene, and so it only have to break the other one for everything to go horribly wrong. So that's usually why these things can be familial. It's not that you have two bad copies to start with, it's just it gets there eventually.
So my first sort of reaction to this question was kind of, nothing. But and that's in part because the way that evolution tends to work is that if a gene is advantageous, there's reason to keep it. But there are checkpoints in bacterial DNA, just as there are in human and eukaryotic DNA, that do very similar things. But I also want to spend a minute here to talk about some like human-directed ways that we sort of keep genes from going rogue in environmental systems. So there are some scientists at a Department of Energy laboratory that is working on essentially they spend a lot of time working on the best ways to sort of engineer microbes that can be useful to the environment, whether that's through remediation or increased crop and biomass productivity. They try and find those sort of solutions. And some of the mechanisms that they use is something called a toxin antitoxin system, is one way they control for rogue genes getting out into the environment, because there's a lot of concern from the environmental systems that an engineered microbe is going to spread, essentially. And so these toxin antitoxin systems they're actually quite natural in some species of microbes, but essentially there are genes that encode both a toxin and its cure, right? So without the antitoxin, the microbe will produce the toxin and it'll build up and kill the microbe. And so it has to be paired with the antitoxin to survive. And the scientists can use CRISPR-Cas9 systems to essentially engineer an on-off switch for these systems so that they can turn the toxin on, turn the antitoxin off, and just regulate and control based on temperature, pH, or things that are normally found in the environment.
So these are toxins for the bacteria, because not for you, not for plants, not for. So these are also called plasmid addiction systems.
Yes, yeah.
So basically, it's like it's in their natural state. Remember, plasmids, you can think of them as parasitic DNA. And the bacteria doesn't necessarily want to have them all the time because they have to go through the energy to replicate them. The plasmid addiction system forces the cell to keep it because the toxin is stable, the antitoxin is not. So when those daughter cells divide, if they don't get the plasmid, well, they still have the toxin and they don't make the antitoxin, and that's a dead cell. So they force the bacteria to keep them. So actually with that in mind, talking about genes going rogue and bacteria protecting themselves, you already mentioned it. So who's familiar with CRISPR? Raise of hands. Okay. Who knows that the early work on CRISPR was funded by Dannon Yogurt? CRISPR is actually part of the bacteria's immune system to protect it from foreign DNA, to protect it from viruses, to protect it from plasmids. And that early work funded by Dannon was because they had something that kept killing their yogurt cultures. Now we use it to engineer human cell lines. So you never know where basic research is going to lead.
Yeah, it was fascinating. They were finding all these little repetitive stretches in the bacterial genomes, and they're like, what the heck is this?
And why does it look like a virus?
Why is this here? And they eventually figured out that when the bacteria were getting infected, they were taking little snippets of the virus and sort of shoving it into its own genome. It has this like little reference library. and is basically like, if I ever see this again, I'm going to kill it with fire. And so it turns out bacteria can remember pathogens the same way our immune systems can. We didn't think that they could do that for a really long time.
They have genetic memory.
But yeah, so that's what CRISPR is in the wild. And it's not the kind of thing anybody found because they were looking for it. I'm going to plug basic research again. This is the kind of stuff you stumble on because somebody goes, there's this weird repetitive sequence in my bacteria. What? And then it's turned into this amazing piece of tech that we can use for all sorts of other stuff. So,
and it's a clunky acronym too. Clustered regularly interspaced repeat.
Something repeats. Genomic repeats.
Trust us, we're scientists.
Thank you.
All right, so as we've been here talking about rogue genes, I've been thinking we're at Dragon Con. This is our celebration of pop culture, and I can think of at least 2 episodes of Star Trek that involved genes going rogue, and there's probably a whole bunch more I can't think of. What are some examples that we see in media of genes going rogue? How accurate are they, and what can we say about them?
Pretty much any zombie media.
Mm-hmm. Yeah. Modern zombie, because it used to be radiation, right? Just genetics are the new
that's still
magic science.
Mutation, right? So falls under similar category.
Yep.
Let's see. So in the 40K universe, there are multiple factions that involve, genes going rogue. Space Marines can consume and gain genetic memory. There's the Kroot, but the biggest would be the Tyranids, which are these giant bug things that will consume all of the biomass on a planet and harvest any useful genetics and incorporate it into their own in some kind of weird biological bug-based Borg-like thing.
I mean, there's almost too many for me to even count. The one that sprang to mind immediately for humans is Dune. You have the Bene Gesserit that have been working for millennia trying to breed people for specific traits. And then you have stuff like The Expanse, where you have alien intelligence coming in from the outside and being like, I'm going to take this thing apart, figure out how it ticks, and put it back together and use it for my own ends for the work. So there's a lot of this in pop culture. And I think actually a fair amount of it doesn't make me wince. I think they actually tend to do their research pretty well on what is and isn't feasible in a sort of a futuristic kind of biomedical setting.
Yeah, I think for The Last of Us, if you guys remember one of the opening scenes of the first episode of The Last of Us, where there was basically a news show where they were talking about the outbreak, that was actually backed by a lot of scientific talk, right, with scientists to say, how plausible is this? Is this the right direction? Can we go? And also, I also want to plug, since the theme song was, I also want to plug Michael Crichton, who was a scientist. He actually dropped out of his PhD program to write books because he was more passionate about that than doing his research.
Michael Crichton doesn't really like scientists, though. That's very clear.
That's fair.
I mean, plenty of people who have been through the sciences would agree with that. Yeah. Let's see. So how accurate? Tyranids are not accurate at all.
Yeah, I think more the thing that bugs me in a lot of pop culture is just the speed with which things sometimes happen, whether it be the spread of the disease or the development of cures. I mean, what was that terrible Dustin Hoffman movie? The Hot Zone? Outbreak, thank you. Right, they capture the monkey, they pull some blood, and within 2 hours they're synthesizing antidote serum.
Ooh,
what?
That would be nice.
Yeah, iZombie was similar. It's all we need is this one blood sample and the plague's over. You're like, that's not how that works.
I think one of my biggest pet peeves in pop culture science media is when they misuse equipment like pipettes as hypospray.
I love that. Yeah,
it's not a syringe, y'all. You use it like this.
And it's always a P1000.
Yeah.
Always.
Well, they're easier to hold. I think the thing about a lot of the way that genes get used in media that bugs me is let's just use the sort of a chronic offender of the X-Men. The X gene it's one gene, and it does all like we just talked about what a gene is. A gene typically makes one thing. So, like, you're not going to get the ability to shoot I-beams from one gene. I'm sorry. It's just not going to work like that. And in particular, there's no reason that one gene's going to do. Are there two mutants that even have the same power? What? Well, I don't. Anyway.
And why not?
Yeah, there should be. Like, they should be, like, super inheritable, right? They are genetic, right?
The one that drives me nuts, and Star Trek does this all the time, is alien DNA. DNA is a really specific molecule. It's that pretty double helix, right? And what are the odds that any other species in the galaxy would have the same type of genetic material? It's pretty much zero. So there is no way you would be getting alien DNA from your crime scene on your swab. There's no way any of these people are having cool half Klingon babies. Klingons do not have DNA. I think Star Trek kind of tried to hand wave it. They're like, the universe was seeded by the forebears back in the day.
Oh,
which is why we all have DNA as our genetic material. But that's the one that makes me always like beat my head on the table is alien DNA. It's like, you could just say alien genetic material and I'd be a lot happier.
Yeah, I like to say you'd have an easier time crossbreeding with a daisy than an alien. Like a much easier.
10 o'clock, monster erotica.
You share 90% of your genome with yeast.
All
Yeast
right.
babies.
So one more thing. You mentioned people being concerned about like genetically engineering microbes and them going rogue and stuff. I've also heard the argument about GMOs in general, about people being afraid, oh, this like herbicide resistance gene is going to go out and make super weeds or other stuff like that. Is there any truth to that we should legitimately be concerned about? Is this like overhyped propaganda? Is there like what are people doing to try to keep these changes under control?
That's a complicated question, Jason, with a complicated answer. Does someone else want to jump in on this?
I'll let you start.
You let me start? Okay. First of all, genetically modified organisms. Another great example of, not in the form that we use it, but genes going rogue. So there's a couple different ways that we make genetically modified plants. One is literally you can load some DNA onto tiny little particles of gold or tungsten and you shoot it at the plant with an air gun. It's called biolistics. And you can take some of those regenerate cells that haven't died but have taken up the DNA, and plants, you know, with the right conditions, you can take one plant cell and grow a whole new plant from it. That's what makes plants really, really cool. You can't take your finger and grow a new hue, but, you know, if you take the right piece of a plant tissue, you can.
And you can't graft me onto my friend either.
That's a different conversation. The other way that we do this is actually based on a plant pathogen called Agrobacterium tumefaciens. So this is a natural plant pathogen. It causes a disease called crown gall. It lives in the soil. And it has one of these systems. We talked about how plasmids can like make little things and send DNA to each other. This bacterium will make a little bridge and ship a chunk of DNA into plant cells. And that piece of DNA codes for a couple things. It codes for plant growth hormones so that the plant cells start growing out of control like those tumor cells, no brakes and all gas pedal. It also gets the plant cells to make something called an opene, which is a special type of carbon and nitrogen source, two of the key nutrients that everything needs, that coincidentally that bacteria can also break down and other things can't. So it turns the plant cell into a little food factory for itself. So what's interesting about that is all that entire system I just mentioned, every single piece of that is encoded by a plasmid. So that bacteria actually isn't really even a pathogen. That plasmid is a pathogen and does all this weird stuff. So what we do as scientists is we take all those genes that make the disease, say, well, let's just take those out. Let's just put our own gene in for, let's say, making glowing petunias, for instance, or purple tomatoes, or herbicide resistance. Put that in. Let the agrobacterium deliver the DNA, regenerate a new plant, and now it's got a new trait, right? So this is how we do it too. Now that's how you get a plant. Now can it escape, right? I mean, for things like herbicide resistance, it is not impossible for that to exit, as far as I am aware. It has happened.
How widespread is it? Just like it's like if it's herbicide-resistant corn, is it just only other corn, or could it spread to like onions and pecans and other things like that?
No, it can't spread. Oh, sorry,
Well,
Topher.
part of the problem is that we don't we often forget that a lot of the plants that we grow for food are all the same bloody species, right? I mean, cauliflower, broccoli, Brussels sprouts, kale are all the same species. So if when we manipulate one, it's going to be trivial for a manipulation to move into the others.
That said, there is no, to my knowledge, GMO Brassica oleracea. There is Brassica rapis, which is canola, that again has traits in it. But yeah, there's really not that many GMOs. I don't know if people realize, like, if you go to the grocery store and you see the non-GMO, I'm sorry,
There's like 12, right?
Yeah, that's it. Like, if you go to the store and like you're gonna find that non-GMO label, like you're really not gonna find your GMOs in the produce section. It's gonna be things like maize. It's things like cotton. And you know what? I don't think we're eating much. I mean, this isn't very tasty. So generally it's row crops. And there's a ton of reasons for that I'd be happy to talk about in a different panel than this for why there's such a small number.
Now, when it comes to engineering stuff in animals, something that's come up a lot now that we've got, behold the magic of CRISPR, is gene drives. And the goal with that, like a big one that people have talked about a lot, is wiping out the Anopheles mosquitoes that give people malaria. Malaria has killed more people than literally anything else in the history of the world. And the Anopheles mosquito isn't necessary for the food web in the ecosystem it lives in. They've crunched the numbers, they've done the math, we could wipe it out, everybody would be fine. And so people have worked out a few methods that they think could work to start passing around things like sterility genes through the mosquitoes. But then the question is, how do you rein it in? How do you make sure this doesn't get loose and turn into another kudzu problem where it then eats the world? And there's a lot of different ways that smart people have come up with to do that. One of the more obvious ones is you put some sort of basically what they're calling, a kill switch in there, where the whole system doesn't work unless it's constantly being exposed to some small molecule, chemical, something that you're constantly introducing into the area. And as soon as you stop, the whole thing grinds to a halt. So
speaking of Jurassic Park, the lysine contingency, right?
Yeah. So there's a lot of ways.
As long as that tune is playing on the island, then they can't escape.
That's right. And so, there's a number of control systems that people have thought about for these kind of things. But everybody has gone back and forth on the mosquito gene drives. I think eventually we're going to do it, but everybody has to be really, really sure that we can control it before they're willing to let it loose.
And I think there's less like general concern of a bacterium DNA that is designed for some sort of resistance in a plant getting, into other plants, right? Because it's, still that whatever that DNA is designed to do, if that's herbicide or pesticide resistance for that plant is still either consumed by people, animals, or made into some other sort of product. So we're still being exposed to it. I think there's a little bit more concern, at least from funding agencies, about microbial or single cell microbes that have been engineered for a function that to be put in the soil getting out. And I think that's more from a patent perspective than it is from an environmental perspective, because when you put a single cell organism that's not native to that environment into an environment, it is very quickly outcompeted by the natural community. So there's not a huge risk for that organism to survive long term in that community.
Yeah, I think this is something that gets lost in the GMO conversation a lot, which is that most of the issues around GMOs are because companies are jerks about IP. It's not because the GMOs are dangerous. It's because the companies are greedy and they don't want people who didn't pay for it getting a hold of their magic seeds that they engineered. So Monsanto is a bag of wieners, but it's not because the corn is dangerous. It's because they're greedy bastards.
To be fair, if I invested $10 million in something, I might want a little bit of return on that too.
Yeah, it's the sort of unintended consequences of economics and regulation. That's why we only have 12 GMOs.
Anyway, so we are now going to open up the questions. Now, do we have a mic runner in here somewhere? There's our mic runner. All right, so some quick logistics on the mic. Go ahead, if you have a question, please raise your hand. Our lovely mic runner will come up here. Some rules about the question: please ask one question. Please keep it short so we can have a lot of people. And most importantly, your question must be given in the form of a question, not a comment.
It's Jeopardy rules.
All right, here we go.
Yeah, eat the mic.
Right. Hello. Oh,
No, you got
okay.
it.
You can hear me. Cool. What about, like, we've talked about rogue genes. What about things like epigenetics and, let's say, people that are famine adjusted or famine adapted, or people who are descendants of, like, big things like a famine, or even like people who are descendants from people that survived the Holocaust, or also, how does that impact into, I guess, rogue genes?
Yeah,
wow.
that's a whole thing.
Yeah, epigenetics is a whole complicated thing that as a geneticist, I try very hard to ignore.
Would somebody like to? Can somebody define epigenetics for the audience?
Basically, there's a whole extra layer of control of the expression of your genes that is called epigenetics, and that is influenced by your environment. So, like, I think one thing people have speculated on, you grow up someplace really hot when you're a little kid, your body adapts to that, and you sort of gets locked in through epigenetics as to how you react to hot versus cold.
There's little like chemical decorations that are put onto your DNA that help reinforce keeping
little
certain genes on or off.
Christmas
Methyl
ornaments.
You guys know how COVID babies are built different? It's because of the stress from COVID. From the mother who was pregnant at the time being under such stress.
I mean, the good thing about epigenetics is it's pretty self-limiting. It runs out in a generation or two. It's really designed to help your body know the kind of environment you're being born into. Are you being born into an environment that's high stress? Are you being born into one that has food scarcity? Are you being born into one where it's 110 degrees every day? And so if that's not true for you when you're born, more of those little methyl Christmas ornaments get knocked off, and it's less true for your kids, et cetera, et cetera. So it really only persists probably through like grandchildren, and then, it sort of readapts for whatever happened to them.
Hello. All right. So, are you guys familiar with the Alien franchise? I wanted to see what your guys' opinion was on the black goo and how accurate the black goo would be to creating life on different planets.
Oh, this is
God.
recent alien stuff, huh? I do think that the idea of an alien parasite really misunderstands how adapted parasites often are to their hosts. Parasites are very highly tuned to their hosts, and you're not going to have something that evolved in a completely different planet that's able to, maybe if it's just a warm, wet space, but aren't they stealing genetics and adapting to that? Yeah, that I don't know, man. That doesn't seem... We haven't met any aliens as far as I know, so what do I know? But it doesn't mimic what we know from biology on Earth.
Okay, next question.
I don't need a
Nope.
mic. Everybody
No,
can-
please
you need
use the
the
mic.
mic.
Okay, fine. All right. So my question is, which environment tends to produce the most mutations that actually allow the cells to survive? So, radiation gardens, back when they experimented with that, or thermal vents, things of that nature.
I think, at least from a bacterial standpoint, any sort of really high stress environment. So yeah, radiation, hydrothermal vents, maybe not the Arctic because that's cold. So they're going to evolve on a much slower basis. But anything where heat and some other stress is applied is going to cause a higher mutation rate.
I want to
In
go into
general.
radiation gardens, but I don't think we have time.
All right, back there.
Hi. Sorry. Straight into the mic. Okay. So back to people go through the Holocaust or a famine or whatever. But what about I don't want to say radiation, but if like my grandfather was in like Japan before Hiroshima kind of thing, and his kids then got something and then I got something, how many generations do you think it would be before that goes away, or does radiation just stick forever for the rogue genes?
Oh, radiation itself doesn't stick around. Radiation basically can cause damage to DNA. That once it replicates that lesion, because DNA is, it is a thing, it is a chemical, right? So when it gets damaged, if it gets repaired wrong during replication, that's what turns into a mutation. The radiation is long gone. If it's an actual damage to the piece of DNA, again, we talked about this, if it's in a body cell, your body cells don't turn into children. So it's got to be a mutation in a germline cell that can turn into either a sperm or egg cell to be passed down at all. But the radiation won't really stick around to DNA. Okay.
Hi. Okay, so zoonoses, super topical right now. What makes them so dangerous, and are there ways of sort of like working around with technology to combat them better?
Oh boy.
Zoonotic diseases. We need to define that first.
Yes.
Zoonotic disease is basically one that arose in another species and then makes the jump, as was said, a lot of these
bat
are
and a
adapted.
pangolin love each other very much and they want to have a baby.
Yeah, they've adapted to attack one species and then they develop a modification that allows them to jump to a different species. So we get bat things that come after us. Can't imagine where that idea came up.
So the SARS virus, which COVID is one of the SARS viruses. I can't remember what SARS stands for.
Severe acute respiratory syndrome.
Yeah, that. Good job.
I lived in Canada when
most
that
of
happened.
those are the zoonotic diseases.
Yeah.
Yeah.
But your question was, how do we make zoonotic? Wait.
Well, I think the first question was, why are they so scary? Because they can just blindside us.
We don't know where the next one's coming from.
Yeah.
Yeah.
And can we?
We can guess.
And the second question was, is there a technological way for us to try to circumvent the threat?
They're kind of a symptom of, climate change definitely is part of that. Climate change definitely exacerbates the situation because it brings wild animals into closer contact with people. It increases heat.
Come to the panel tomorrow at 1 p.m.
Yeah, it's there's no simple answer to that question.
Yeah, a lot of it is.
But
we're also moving a lot closer into these urban wildland interface areas. That is part of a cause of this. Is that we're invading their territory and they're going to invade ours right back.
I would say, at the risk of straying into politics, investing in public health, investing in disease surveillance, like what the CDC and the WHO do. Those are good things for catching these bugs.
Yeah, it's not gonna hurt.
I was going to say there's a whole field of science devoted to it called epidemiology.
Yeah. So you talked about adding and removing DNA and.
You don't need to look at us. Look at the mic.
Yeah.
So you talked about adding and removing DNA. So this is kind of a ships of Theseus question. Is at what point do you consider the cell to be a whole different cell than the original through modification?
Good question.
It's basically speciation, isn't it?
Yeah. But so for bacteria, when is it speciation? Because bacteria are all like 99.9%. Well,
Not
they're
all.
really similar compared to like the difference between a human and a chimpanzee. Like you can tell in so 95% similarity between a human and a chimpanzee, right? And you can tell by looking that they look different. When you get that 95% similarity difference between, or not 95 difference, a 5% similarity difference between between two bacterial species, you can't tell by looking at them. You have to look into their DNA to tell that they're different. So when is speciation?
This is a fuzzy question, even in like birds. So honestly, species are all made up.
Wait,
Thank you for coming.
Birds are fuzzy?
Yeah, I was gonna say this is who knows what a species is, because I would love to find out.
That question, what is a species, is still in debate by scientists.
Yeah.
Okay. Next.
Hello.
Hello, you appear to have some rogue genes on your
Right.
forehead. I'm sorry.
Sorry. Okay. This is kind of an ethical thing, but you were talking about CRISPR, and I think it's really interesting. But at what point does it become like unethical to use? Because I hear a lot about like designer babies, and I don't know if that's just something like way out there or actually like.
Yeah, no, there's been a lot of discussion about that.
Yeah, and do you think that it might be an inherently eugenicist process, regardless of whatever connotation that holds? Because I don't, yeah.
So generally speaking, the scientific community has talked a lot about this. Obviously, this is a hot point of debate. And as a rule, people have come down on the side of we are cool with using genetic modifications like CRISPR to help cure and treat disease. We are not cool with using it to make Captain America. And I think most people, you do public opinion surveys, I think most people agree. We're generally not okay with things that are cosmetic changes, with things that are intended to make somebody smarter or good at music or that kind of stuff. People are kind of like, I'm not comfortable with that. But over on the side of we would like to cure, multiple sclerosis and inherited cancer syndromes and inherited blindness, people are like, sure, great. The fuzzy area, of course, is the bit in the middle. And there are things where a cure in somebody with muscular dystrophy, if you do the exact same treatment to a healthy person, you end up with Arnold Schwarzenegger because they bulk up. So there are things that are both treatments and also in a healthy person would be a basically
Enhancement.
a performance enhancing drug, essentially. Athletes use EPO. It was developed originally to treat people who had low blood cells for other reasons. But anything can be used both ways. So that is the line that we're on right now. And the NIH has come down on the side of it has to be used to treat a disease that is a serious disease. So things like blindness, something that's going to cause early death. It has to be a disease for which there is not other good treatments available. And you have to be pretty confident that the change you are going to make is going to fix it.
The other thing is that we don't do this, we do this in body cells. We do this in the cells that are not inherited. By getting treated with CRISPR for these diseases, you're not passing that on to offspring because that only happens in egg cells and sperm cells.
You touched on inherited blindness, and that is actually a gray area, because in blind communities, in deaf communities, there's an entirely unique culture that arises, right? If you have two deaf parents raising a child, they may very well want their child to be deaf because it's going to face the world in the exact same way they do. And so treating inherited, I guess my point is that blindness is a great example of one of those conditions where is it a disease or is it just, a variation of humanity?
Yeah, and I think that's a call that is up to individuals and parents to make. Yeah. We make all kinds of decisions on behalf of our children and our unborn children all the time, and this is another one of those things that's going to get added to the list.
Yeah, but the ethics is freaking complex.
All right, we have time for one short question.
Sorry, I know you said one question. My question has sub-questions. Just about...
That is multiple questions. That is not one question.
Okay, so then just one question about precision medicine. Do you think precision medicine can be used to maybe target drug-resistant bacteria, antibiotic-resistant bacteria, or not?
In short, yes. So precision medicine is it's a really broad area. It can mean taking a look at someone's genome and designing medicine that works specifically for their genes, or it could also mean taking a targeted approach with labeled enzymes, labeled proteins to target organisms and disease. It's being used, that label approach, the isotope labeled approach, is being used for targeting cancer in studies now.
Okay, all right. That is all the time we have. If you want to talk more, you're welcome to come up here. Before you go, though, panelists, where can people find you? Let's start down with Jennifer and work this way. Either here at the con or afterwards, social media or whatever.
So I'm going to be hanging around here as long as y'all have questions for me. But I don't actually have an Instagram or a podcast or anything. So you're going to have to hunt me down here and now.
You can find me and Jason too at gamingwithscience.net. And we've got a bunch of socials that use that same tag. Instagram, we do all the things.
And you can find me on socials at anisotoph, ANISOTOPH. And I'm on a whack ton of panels, so just look me up in the app.
The only social that I really monitor is LinkedIn, so you can find me on LinkedIn. Or if you want to hunt me down at the con, I'll be here on Saturday for the Science Power Hour. On Sunday, I am on the Slime Time panel, and on Monday, I'm on the What You Didn't Know the Government Pays For panel.
All right, and I will be here, I believe, in this room tomorrow morning with WTF is Grass. And then in the ballroom tomorrow afternoon for Dr. Robotnik's Guide to World Domination. If you want more science track today, then the next one here is at 5:30, which is the Science of Propaganda, and in the ballroom at 8:30, we will have Silly Science and Remarkable Results. And thank you. Please remember to fill out the feedback. We do use that. That is not feeding an algorithm. We people, like human beings, actually read that, and we take it into account for making the content. Donate to the charity, and have a great Dragon Con. Thank you so much for coming out.
We're human beings. We're human beings.
No, probe. I can't believe I fist bumped you.